Okay, so let's finish up chapter 24 today. Now, we're going to go over some different prokaryotic cells, just basic overview, nothing too intense. But before we go over them, we're going to talk about just prokaryotic radiation. So in other words, we started off as one group of prokaryotes, and they went through many different forms of divergent evolution in order to have as many prokaryotes as we have today. So first big one was we were splitting these prokaryotes into two different groups. We were splitting them into bacteria and archaea. And then later on down the road, we also then began to have eukaryotic cells. Oops. Okay, so we talked about horizontal gene transfer in a previous chapter. There was horizontal gene transfer and vertical gene transfer. And when we talked about vertical gene transfer, this was transferring of genes from parent to offspring. Now, horizontal is going to be during your lifetime, your DNA changing. So what we're doing here, horizontal gene transfer, what's going to happen for our prokaryotic cells is, I know we talked about conjugation last time. When we have conjugation, when we have one bacterial cell and another bacterial cell, and we have that pili connecting them in the DNA transfer, this is going to be a form of horizontal gene transfer in our prokaryotic cells. All right, so here are our three, and I just showed you guys a picture of this earlier, but our three different main types of domains. We have bacteria, archaea, and eukarya, and the bacteria and archaea split first. Now we're going to go over some of these different groups here. First one, proteobacteria. So this is a gram-negative, which means they don't have a whole lot of peptoglycan. It's going to be our photoautotrophs, our chemoautotrophs, and some heterotrophs. Some of these are good with oxygen. Some of these are not good with oxygen. So in this slide, guys, I talk about the mitochondria. I'm not going to focus too much on alpha proteobacteria, but I do want to talk about the mitochondria. So the current hypothesis is that the mitochondria rely on horizontal gene transfer for the movement of their genes. What was going on was their genes were once present in their chromosomes, and these genes moved to the nuclear genome. In other words, it moved to the nucleus. Now, don't forget, mitochondria, they are in eukaryotic cells, so we're talking about a eukaryotic cell now. But this is why we say mitochondria originally had their own DNA, but now they live in eukaryotes, so they kind of transfer that DNA to that eukaryotic cell. All right, so here's some other ones we're going to talk about. First one is chlamydias. And I know you guys are probably thinking like, oh, I've heard of chlamydia before. We learned about it in health class. It was like some kind of STD or something like that. Yeah, not all of them. It's just, this is a general term for some parasites that live in our cells. So they're not always related to sexually transmitted diseases. I don't want you guys to think that. But anyway, the one we're going to go over is called trachoma. So this is a disease or illness caused by chlamydia, a certain type of chlamydia. But this is when your eyelid can actually turn inward and your eyelashes will scratch your cornea in your eyes. This causes like serious pain. And if it goes on for long enough, it can result in you pretty much going blind in the end. So completely different. You're like, oh, that's not the same as the STD chlamydia. No, it's not. So completely different effect on your body. Next one we already talked about, cyanobacteria. These are our early photoautotrophs. They generate oxygen. This is what helped the oxygen levels get to where they are at today. There's also going to be some gram-positive bacteria we're going to go over. I'm not going to harp on these too much, but there's a gram-positive bacteria, and here's another one. These are taken with scanning electron microscopes. Okay. Now, if we were to stain them, we would see that they would be a purple color. Now, archaea. These are going to share some traits with bacteria and other traits with eukaryotes. But our archaea are actually more closely related to eukaryotes than bacteria. Kind of like fungi. You know how I said fungi is not a plant? Fungi is actually more closely related to us than it is plants, based on the nature of that organism. But archaea, more closely related to eukaryotes, us, than bacteria. So this means that a lot of its structures, like the ribosomes inside of it, are actually going to be the size of eukaryotic ribosomes, larger, than the bacteria ribosomes, which are going to be smaller. Archaea actually does not have a cell wall either, but they do have a plasma membrane just like bacteria. And just like bacteria, they also don't have a nucleus, but they don't have that cell wall. So that's something that they don't have in common there. And here's a comparison, guys. You guys can look over all this, but with our bacteria, archaea, and eukarya, and just the differences between them. Here's our second one here, just with the type of DNA and things like that. But you guys can see that the circular DNA is actually present in archaea and bacteria. So the DNA is probably closer to bacteria with archaea compared to eukarya. All right, so touching on archaea, we already talked about they live in extreme environments. So there's two different types of archaea. There's extreme halophiles and there are extreme thermophiles. So extreme halophiles will be fine in a very, very high salt content environment. So they will be fine living in a very hypertonic environment. Because if there's a lot of salt in there, obviously the environment would be hypertonic to the archaea, which would be hypotonic. So what happens here is they naturally produce, in order to live in this hypertonic environment, they naturally produce a salt-like product in their body in order to counteract that high salt environment, that high hypertonic environment, and that allows them to be fine. The extreme thermophiles, what they're going to do is they are going to have certain enzymes that can function in these hot environments in order for them to survive. And there is a picture of these extreme environments that the archaea live in. Okay, so our next one, Methanogens. These are archaea that are going to use CO2 to produce methane. And methane is a waste product, so they don't want to keep methane in there. They're going to get rid of the methane. So in swamps, marshes, places like that, these are going to be areas that are very high with methane. Cattle, very high with methane. If you ever heard of, sorry to bring this up, but if cows have gas, you can actually light them on fire because of the methane. So they actually do have the methanogens that live inside of them. Yeah, that's why it's like that. Okay, we're good here. All right, last section. We're going to talk about prokaryotes and just their role on Earth. So when we're talking about the role that prokaryotes play, if we did not have prokaryotes, we would probably not be around today. There's a very good chance that we would not be around. So most of these prokaryotes can actually decompose things. Now, we're not just talking about like, oh, look, a tree fell down. Let's get a prokaryote there to decompose it. No, not just that. Dead organisms, they decompose. Waste products. When we go to the bathroom, they decompose all of this stuff and then put it back into the environment. They essentially recycle everything. So along with this, they can increase, with recycling, they can increase some of the elements that we need for our bodies. Nitrogen, phosphorus, potassium, okay? Things that we need, things that plants need. So as they decompose, they increase the availability of these elements. And what's nice about this, guys, is if they don't need it, they're just going to leave it. So they know what they need, they know what they don't need, and if they don't need it, they're just going to, okay, well, I don't need this, I'm just going to leave it over here. So our last thing for the chapter, guys, is on symbiosis. Now, symbiosis is some type of relationship between two organisms. Now, in the past, and even you guys talked about symbiosis, you're like, oh, it's when two organisms benefit. It is, but that's a specific type of symbiosis. We're going to go over those 3 different categories where it's not always benefiting both groups. So our first one is going to be mutualism, and that's the one you guys know. Both of them are going to benefit. Second one, commensalism. What's going to happen here is one organism benefits while the other is just chilling. All right, it doesn't benefit, but it doesn't get harmed as well. The last one is parasitism. This is when we have a parasite, essentially. It's good for one, the parasite, and it's really bad for the other one. In other words, we would call that the host. All right. Now, the goal for the parasite is not to kill the host, because if it kills the host, then it has to find another host. Okay. If it can just thrive off of that host, it's cool. So it doesn't want to kill it. So here is the remora with a shark there. This is an example of commensalism. If you guys have ever seen the remoras swim near the shark, what they're doing is they are getting protection from other animals, and they can actually cling to the shark where they will get a ride. Now, in turn, they're not harming the shark at all, but the shark doesn't get any benefit from it. So this is commensalism, the shark and the remora. This one here, we have this fish, and if you look near its eyes, it has a bioluminescent bacteria there. And what's going on is the fish can actually see better way, way, way down in the depths of the ocean. In turn, the bacteria gets a nice place to live. Okay, this is mutualism. So both are benefiting, no one is getting harmed. Parasitism, guys, I'm not gonna give you pictures of that one. One thing is harmed and one thing is not harmed, okay? That'd be like getting like a staph infection or something like that, We don't want to explore those routes again. All right. So best known prokaryotes are pathogens, but we can actually have ones, and pathogens are, bad, but we can actually have prokaryotes that are really good for us. And most of them are positive interactions. Most of them are not negative. Our intestines have so many different bacteria that help us break down things. What they do is they help us with our digestion process. And if we didn't have them, our body would be using a whole lot more ATP in order to digest our food. And if we were using more ATP, we would be more tired throughout the day. With our prokaryotes, guys, they do cause about half of all of our human diseases. The example your book gives is one on Lyme disease. That is a disease carried by ticks. If you guys have ever seen, sorry, the bell. If you guys have ever seen this kind of thing, all right, you might have gotten bit by a tick with Lyme disease. Symptoms, you could have pain, you could have mood changes, joint pain, vision changes. Your skin might have some outbreaks. You might have rashes, fatigue. You might have trouble sleeping. Seems like a lot of normal things, though, Like, that could be that, or you could just have the common cold, So, if you think you got bit by a tick, or if you're outside and ticks are really prevalent spring, summertime, when you come in, you might want to check and make sure you don't have any ticks under your skin. The thing about a tick is it actually injects something into your body before it goes in that is like a novocaine, like a numbing agent, so that it can bury into your skin without you knowing it. All right, so with these bad bacteria, with these bad prokaryotes, we have two different categories. We have exotoxins and endotoxins. Exotoxins, they are released anywhere. So they will be released outside of the prokaryotic cell. And yeah, that's bad for us. Endotoxins, these are only going to be released inside of the bacteria when it dies. And it's going to be released inside the cell wall, and it's going to break that cell wall down. So last slide today, guys. We already went over horizontal gene transfer. Okay. Now, with horizontal gene transfer, and we saw this in the experiment with the R strain and the S strain from chapters ago. If you guys remember, the R strain was the good strain, the S strain was the bad strain. When we mixed, here, I'll write it down for you guys here. So, our R strain was good bacteria. In other words, inject that into you're fine. Our S strain was our bad bacteria. In other words, inject that one into you and you will die. And they found that if they heated the S strain bacteria, it pretty much killed the bacteria, and the bacteria did not kill us as long as it was heated. But they also found if they mixed some heat-killed S strain plus some regular R strain, that it would die. But each one of these things separately, no problem. What happened was the S strain DNA actually conjugated over into the R strain by horizontal gene transfer, and it made that R strain become S strain. In other words, it made the R strain become that bad bacteria. Okay, so that is a way horizontal gene transfer can spread virulent or bad bacteria, things that can cause us harm, even if that S strain or bad strain has already been killed. Okay, guys, so that is the end of chapter 24. We'll have a quiz on this, and then we will have your unit quiz, and we are done with quizzes for the year, and then we will start on the review before your AP test. Have a good rest of your day, guys.